Let me know in the chat if the audio and video are clear. Let me know, guys, how was your bio paper? We are going to be discussing questions from set 3. If everybody is ready and you're anxious to find out if you've answered correctly or not, let me know in the chat and we'll get started. Good afternoon, everyone. Is the audio and video clear? Okay, so let's get started. We'll start off with the botany portions. I will discuss the botany questions and later Vishwajit sir will take over and discuss your zoology syllabus. You got set 2. We're going to be discussing questions from set 3. Okay, so we also have JEE and NEET channels where we coach in English, similar to the PW English medium channel. So the names of the channels are given here. In case you're also aspiring for JEE or NEET in the coming years, you can consider subscribing to our channels. So we'll get started with the first question in set 3. So here, oh my god, why is this not writing? Just a minute. Never mind. I'll just tell you the answers. Okay. Perisperm present in the seed of black pepper is remnant of ovary, nucellus, anther and megaspore. Perisperm is something that you find inside of seeds like black pepper and beet, and that is the remnant or residue of, remnant of nucellus. A thin layer of nucellus persists even in the mature seeds and you call that the perisperm. So the correct answer to this question is option B, nucellus. For some reason, I'm not able to use my pen, so I'm not able to tick it. But remember that the correct answer to this question is option B, nucellus. Now, moving on to the next question. Which of the following sacred groves is found in Meghalaya? This is a question from the chapter Biodiversity and Conservation. In the options, we have Jaintia Hills, Bastar, Chanda and Sarguja. So the correct answer is Jaintia Hills. This is what we find in Meghalaya, Khasi and Jaintia Hills. And Bastar, Chanda and Sarguja, these are found in Madhya Pradesh. Right? Next. In an ecosystem, different species occupy different levels and vertical distribution of species is found. What do we call this? This is a question from the chapter ecosystem. So in the beginning, we are told that there is a physical structure for each ecosystem, and one example for that is what you can find in forests, right? So in forests, you find different plant species occupying different vertical zones and we call that stratification. So where is stratification? Yeah. Option A is the right answer to this question. Okay. Moving on, what do we have next? Which of the following statements about plasmids is incorrect? We learn about plasmids in the chapter Cell the Unit of Life. We also learn about plasmids in the biotech chapters. So I'll just go through these options and we'll find out which one is incorrect. First option, plasmids have the ability to replicate within the bacterial cell. Of course they do. They have their own origin of replication because of which they can make additional copies of themselves. Their replication is controlled by chromosomal DNA. Okay, so this is the incorrect statement. Chromosomal DNA does not control the replication of plasmids, regardless of whether the chromosomal DNA is replicating or not. Plasmids, since they have their own origin of replication, can can replicate many times, and per bacterial cell you can have many plasmids present. Okay, so option B is the incorrect one here. Option C, they are autonomously replicating circular extra chromosomal DNA. Yes, that is correct. That's in fact how we define plasmids. They often carry antibiotic resistance genes. That is also true. You've seen that with respect to PBR322, they have ampicillin and tetracycline resistance genes, right? So A, C and D are correct. B is the incorrect statement. I'm sorry, I'm not able to mark on the board because for some reason my pen isn't working today. But nevertheless, we'll move on. A DNA molecule is 140 base pairs long. It has 20% thymine. How many guanine bases are present in the molecule? Now, what do I do here? I will have to write. How do I write? Just give me a minute. Let me see if I can use a pen in some way. Oh yeah, I can. I've figured a way. No problem. Cool. So for as long as this thing is there, we can use it. So DNA molecule has 140 base pairs. That is 280 bases in total. Is this visible? Let me increase the size of the pen. So it has 280 bases. Of that, 20% they are saying is thymine. We know from Chargaff's rule that in double-stranded DNA, you adenine plus thymine plus guanine plus cytosine will be equal to 100%, which in this case is 280. T they have said is 20%, so A is also going to be 20%, right? So 20% of 280 is how much? 20% of 280. My math brain is very weak, so I'll have to use my calculator. I hope you guys did not have to struggle in the exam. So let's do this. Yeah. So 20% of 280 is equal to 56. So A is equal to 56, T is also equal to 56. 56 plus 56 plus, we do not know what G and C is, but we know G and C are equal. So let's just take it as X plus X. 56 plus 56 is how much? 10, 11. 116 plus 2X is equal to 280. So X is equal to 280 divided by 116 divided by, minus 116 divided by 2. 280 minus 116 is equal to 164 divided by 2 is 82. Ouch. Where did I make a mistake? 6 plus 6 is 12. No? Wow. This has to be 100 and 102. So 280 minus 112 is equal to 168 divided by 2 is equal to 84. So they've asked for how many guanine bases are present? The number is 84. So option B is the correct answer here. Right? Oh, okay, Tanay. Thank you. I'm glad it helped. All right. So we'll move on. Now that I've figured a way to navigate through this. Oops. Okay. So that 84 is the correct answer. We'll move on to the next question. Select the mismatched pair. Megadiversity countries of the world is 12. Yes, that is correct. There are a total of 12 megadiversity countries that have been declared across the world, of which India is one. Genetically different strains of rice in India is less than 1,000. Not really. We have more than 50,000 strains of rice. So 1,000 is a number that we use for the strains of mango or the varieties of mango. In the textbook, more than 50,000 you have rice strains or varieties. Robert May estimated estimation on global species diversity 7 million. That is correct. Zoological parks is a type of exit to conservation. This is also correct. So what is mismatched here is option B. Okay. Now let's move on to our next question. It's an assertion and reason type of question. Assertion statement reads, in terrestrial ecosystem, much larger fraction of energy flows through grazing food chain. This is an incorrect statement, right? It's wrong, incorrect. How do I use? Yeah. So this is incorrect. This is incorrect. Okay. And okay. And grazing food chain may be connected to detritus food chain at some levels. That is correct. Assertion statement is wrong because in terrestrial ecosystems, major conducive energy is not grazing food chain. Energy flows mostly through detritus food chain. In aquatic ecosystems, it is through grazing food chain. So assertion reason is wrong. Assertion statement is wrong. Reason statement is correct because many a times you see that there is an interlinking or intersection of grazing food chain and detritus food chain. Whichever organisms are found in detritus food chain, they are prey to the organisms in grazing food chain. So there will be some sort of intersection. So here assertion is false but reason is true. Option D is the correct answer. Next question. Assertion statement reads, flocs are masses of bacteria associated with fungal filaments in secondary sewage treatment. Yes, that is correct. That is how you define flocs. You get it in the aeration tank, right? After, you you release the primary effluent in the aeration tank, and then bacteria and fungi will start growing and increasing in number by using the organic matter, and they eventually form what we call flocks that will later settle down as activated sludge. So that is what flocks are, masses of bacteria associated with fungal filaments. They are formed during sewage treatment. That is correct. Reason: flocks help in digestion of solid waste by anaerobic respiration. Not really. All of these bacteria and fungi that you find in flocks, they break down the organic matter aerobically, which is why we pump oxygen, or you make sure that there is proper aeration in aeration tank, because these organisms require oxygen. When they don't get sufficient oxygen, they will not be able to efficiently break down the organic matter. So here assertion is true but reason is false. So option C is the correct answer in this case. Moving on to our next question. How does a farmer use dormancy of seeds to his advantage? See, if a property called dormancy did not exist, we would not have been able to store our seeds. What happens during dormancy? The seeds are not germinating, they are inactive. Just whatever metabolic activity is required for the seeds to remain alive, just that much metabolic activity will take place, and a lot of dehydration happens, and the seed does not contain lot of moisture inside. Because of this property of dormancy that seeds show, it's possible for farmers to store their seeds and grains for a long period of time, and they can use it over the next season to cultivate their crops. If only seeds did not have this property of dormancy, what would happen? Immediately after the seeds were formed, they would germinate. So you wouldn't be able to store seeds for the next season. Especially farmers wouldn't be able to store it for the next season. Like we store grains and seeds at our homes, right? We have pulses which are seeds, and then we have grains like rice, wheat and ragi and all of that. We're able to store them because of this property called dormancy, and that is what even farmers kind of use. So that is the first answer to the first question. Second question, differentiate between a pea seed and a castor seed. Both pea seed and castor seeds are dicot seeds. The difference between them lies in the fact that in pea seeds at maturity, you do not find any endosperm remaining. Whatever endosperm would have formed is completely used up during embryo development. However, in castor seed, even in the mature seed, you can find some amount of endosperm still remaining. So we call the pea seed as a non-endospermic or ex-albuminous seed, whereas you call the castor seed as an albuminous or endospermic seed. So in castor, reserve food is stored in the form of endosperm, and in pea plant, reserve food is stored in cotyledons, in their two fleshy cotyledons. Okay, so that's the difference between pea seed and castor seed. I hope you have answered this question correctly. Next question. Mendel published his work on inheritance in 1865. But it remained unnoticed for over 3 decades. That's true. He published his work in 1865. But when was it rediscovered? 35 years later in 1900, 3 people independently rediscovered his work. Right? Carl Correns, Van Schermack and Hugo de Vries, I suppose. So these guys rediscovered his work. For 35 years, why was his work not recognized? What are the reasons for that? According to your NCERT, you have 4 reasons why Mendel's work did not get the recognition it deserved while it was published. Firstly, it was so early on in the day and communication was a problem. It was not like today's day. So it was not easy to publicize his work. Okay, so not a lot of people got their hands on his work. So that is our first primary reason. The second reason is that Mendel used mathematics to understand biological phenomena, which many biologists back in the day couldn't kind of wrap their heads around. They could not understand it. That is the second thing. Thirdly, he said that there are discrete units called factors which are getting passed on from one generation to another. But because in nature you see such continuous variation, not many people were aligned with the fact that it can be unchanged discrete units like that. If it was that case, then how do we see so much variation in nature is their question. Then fourth point is that he could not provide physical proof for the factors. He told the cells have factors and it get passed on through the gametes. All of that he mentioned, but where are these factors? Show me the physical proof of factors. He could not provide that. So that is the fourth point. Okay. So these are the reasons why his work was not recognized. There are 4 points. You're expected to write 4 points. So I hope you've answered this correctly. Moving on. Okay, yeah. Biodiversity conservation is crucial for maintaining ecological balance and ensuring the survival of future generations, of course. You know about this Rivet-Popper hypothesis. Biodiversity is very, very important. Whenever species go extinct, biodiversity comes down. Then obviously the entire stability of the ecosystem can get compromised. So yes, it is important to conserve biodiversity. Describe 4 advanced ex situ methods used for conservation of biodiversity. Firstly, even in animals, we use in vitro fertilization and embryo transfer whenever the numbers of these organisms or the number of species is very, very low. IVF and ET, embryo transfer, can be used to conserve other species also. Secondly, in plants, they do micropropagation or tissue culture. Gametes and embryos, seeds can be cryopreserved. Uh, Cryopreservation is preserving, storing gametes and all of that at minus 196 degrees Celsius in liquid nitrogen. Apart from that, we also have like seed banks and other sorts of things which can be considered, or germplasm conservation. All of that can be considered as advanced ex situ conservation strategies. Okay, now moving on. We have to wait till the questions are added. Okay, just give me a minute. How was the paper? Let me know in the meantime. Did you all answer well? How much marks are you expecting? Zoo and botanical gardens, of course, are ex situ conservation strategies, but they are not advanced ex situ conservation strategies. So here they have specifically asked for which are the advanced ex situ methods. Advanced means you have to write whatever I just mentioned, like cryopreservation, in vitro fertilization and embryo transfer, all of that. Okay? Just a minute. For some reason, हेलो मैम, ये सेशन एंड हो गया है यूट्यूब की तरफ से या कुछ हुआ है? आई कांट सी द वीडियो. कैन यू प्लीज चेक? आई एम ट्राइंग टू कॉल शाहरुख एंड एनी डेस्क पर्सन, बट बोथ ऑफ देम आर नॉट आंसरिंग.
यस मैम.
सर, ये लाइव एंड हो गया है क्या? देखिए एक बार प्राइवेट.
वही देख रहा हूँ मैम.
हाँ,
मैं भी वही देख रहा हूँ मैम. मैं भी वही देख रहा हूँ. 1 मिनट रुकिए मैम.
ओके ओके सर.
मैं रिफ्रेश भी कर रहा हूँ पर ओपन हो नहीं हो पा रही मैम फाइल ये. जैसे इन्होंने प्राइवेट कर दिया, इसको एंड कर दिया।
क्यों सर ऐसे सडनली?
हाँ जी मैम, जब वीडियो इस प्राइवेट कर दी मैम। ये 1 मिनट मैम बात करता हूँ मैं वाई टीम से।
ओके
जी
ओके।
मैम, है ना।
हाँ
मैम वाई टीम कौन है? गौतमी है या अपनी प्रेरणा ही है?
नो, शाहरुख है।
मैम मुझे नंबर शेयर कर सकते हैं आप उनके?
1 minute sir, I think he's
जी।
saying that it has become proper now.
हम्म।
uh, I'm not able to see it. ये OBS की तरफ से कुछ
ओबीएस तो नहीं मैम, जो वाई टी से ही लिखो।
okay,
या
let
तो
me
उन्होंने एंड कर
check.
दिया इसको, या
Hmm.
फिर इन्होंने प्राइवेट कर दिया। बट वो नहीं होना चाहिए, लेकिन क्लास लाइव था।
हाँ हाँ हाँ, I'm checking sir, just a minute if I can see it. No, I'm not able to see it.
मैं करता हूँ मैम, शाहरुख सर से मैं करता हूँ बात। मेरे को
Okay,
नंबर मिल गए हैं।
okay, okay sir.
मैम आपका पूरा ऑडियो में आपको रहा है जो आप शाहरुख से इंटरेक्शन कर।
Oh, is it? Oh, but it's ended, like I can't see that video. You can see me? But I think because you have the link or something, you're able to see it because I can't see it. It's yeah, just let me know what I have to do. Yeah. हेलो।
मैम आप लाइव है ना?
हाँ, ये कुछ is it live right now? Because the video had become private. Abhi, okay, okay. I will do it. I will continue then. Hee hee. I think you all got a little bit of drama because of the glitch that happened. Is everybody back online? Am I visible? Am I audible? Let me know. All right. So we had finished. We were talking about how Mendel's work was not recognized. I gave you 4 reasons as to why Mendel's work wasn't recognized. Right? We'll move on with the next question. Is everything clearly visible and audible? Just let me know in the chat. Just confirm in the chat so we can move on. Okay. All right. So we'll continue. Cool. So we'll move on to the next question. This is a question from the chapter Ecosystem. What is primary productivity of an ecosystem and how is it expressed? Primary productivity of an ecosystem can be defined as the rate at which biomass or organic matter is produced by plants through the process of photosynthesis. It can be expressed either in terms of energy or in terms of biomass. So you can express it as grams per meter square per year or kilocalories per meter square per year if it is in terms of energy. Second part of the question, explain the equation given below. NPP is equal to GPP minus R. In this case, NPP stands for net primary productivity. GPP stands for gross primary productivity. And R stands for respiratory losses. So, the entire biomass that plants produce by photosynthesis, the rate of that is gross primary productivity. Plants utilize some of that for their own growth and development. So, some of the biomass that is synthesized by plants undergoes cellular respiration, which releases energy, which the plant will use to grow and develop. So, not all of the biomass that is accumulated by the plant is available for us, the consumers. So, gross primary productivity is the total amount. Respiratory loss is something that the plants will use for their growth and development. What remains for the consumers is what we call the net primary productivity. So, the available biomass for the consumers is what we call net primary productivity. I hope that you've answered this question correctly. Let's move on to the next question. Oh, I think we discussed this previously. I gave you 4 advanced ex-situ strategy methods. Here is our next question. Do all pollen grains remain viable for the same length of time? Support your answer with 2 suitable examples. Pollen grains viability period is basically the period during which it retains the ability to germinate and form a pollen tube when deposited on the compatible stigma, right? So in all species, in all families, it's not a fixed amount of time, it's not the same length of time. In some of the species like wheat and rice, usually these grasses, Poaceae members, they will lose their viability within 30 minutes after the pollen grain has been dispersed. So that is our first example where before 30 minutes the viability is lost. The second example that we can give, if you've written Rosaceae, Solanaceae, Leguminosae, any of those as an example for pollen grain retaining their viability for many months, you could have written any of those 3 examples. For low viability period, wheat, rice or grass family, you can write under 30 minutes. Viability period retained for up to many months, that is in Rosaceae, Solanaceae and Leguminosae, these 3. Okay. How are pollen grains stored in pollen banks? Pollen grains are stored in pollen banks at minus 196 degrees Celsius in liquid nitrogen. We call this cryopreservation. I hope you have answered this question correctly. Right? Have you? Let me know. Next question. What is pedigree analysis? Mention its importance in human genetics. Pedigree analysis is a type of analysis where we study any specific trait. It could be a disorder, it could be any trait using certain symbols. Okay, so family chart or across many generations you represent in the form of certain symbols that will give us an idea about how a particular trait is passing on from one generation to the next generation. That is the use of pedigree analysis. You will get to know what is the inheritance pattern that particular trait is following. We will get an idea of whether that particular trait is autosomal recessive, autosomal dominant, X-linked recessive, Y-linked, whatever the case is, we will get to know when you look at the pedigree chart, right? So that is its importance. Analyze the following pedigree and write the pattern of inheritance. If you look here, the parents are not affected in the first generation. In the second generation, this couple has 1, 2, 3, 4, 5 children. Of the 5 children, 2 children are showing the trait. It could be a disorder, it could be any trait. Parents themselves are not showing it, but these 2 kids are showing it. One boy and one girl is showing that trait. If parents themselves are not showing that trait, but the children are showing it, even in this case, parents don't have it, but one son has it, then it is definitely a recessive trait. Now the question is it X-linked recessive or autosomal recessive? If it is X-linked recessive, if the female is showing that trait, then 100% father also will be affected. But in this case, female is showing the trait, father is not affected. That means it is autosomal recessive trait. Okay? Now, this could be something like sickle cell anemia, that is an example. Thalassemia is one example. Phenylketonuria, all of these are autosomal recessive traits. So, give one example they have shown. Example of any one disease. If you write phenylketonuria, sickle cell anemia or thalassemia, all of them are examples for autosomal recessive disorders. Okay, any one of those if you've mentioned, you're safe. You will get full marks. Next question. Draw a neat diagram of megasporangium of an angiosperm and label any 6 parts. Megasporangium is nothing but the ovule of angiosperms. In angiosperms, the ovule is inverted, right? So you draw a question mark of sorts like this. Here is your outer integument. Inside that you have the inner integument. So you will label outer integument and inner integument. The stalk that you have here is called the funicle. The point of attachment of the funicle to the body of the ovule is called the hilum. You have outer integument, you have inner integument. Inside that you have the nucellus. Here you have a small opening that is called the micropyle. Within this you can show a structure called embryo sac and label the embryo sac. This pole of the ovule is called the chalazal end. This pole of the ovule is called the micropylar end. I hope you have drawn this neatly and labeled whatever 6 parts you could remember. So this is the funicle, this is your hilum, this is your outer integument, this is your inner integument, this is the embryo sac or the female gametophyte, micropyle. If you have labeled 6 of these parts, you will get full marks. Moving on, what is our next question? The diagram below shows the distribution of 2 barnacle species. Which are the 2 barnacle species? The 2 barnacle species are Cathamylus and Balanus. Balanus is the competitively inferior larger barnacle and Cathamylus is the competitively the Balanus is competitively superior, Cathamylus is competitively inferior. So what they've given us here, when Balanus, the competitively superior larger one, was removed experimentally from the coast, then the Cathamylus, which was competitively inferior, spread across a more geographical region, right? So when experimentally removed, Cathamylus expands its range in lower intertidal zone. Okay. Identify and define the ecological phenomenon demonstrated by this observation. What does this demonstrate? This demonstrates competitive release. What does competitive release say? Whenever two species are competing for the same resource, one will be superior, one will be inferior. If you experimentally remove the superior one, then the inferior one will spread across in that range. Okay, so that is an example for competitive release. Moving on to the second part of the question, state the principle that explains the elimination of one species. Here they are talking about Gauss's competitive exclusion principle that was proposed by Gauss. What does he say? Whenever two closely related species are competing for the same resources, they cannot coexist indefinitely. Sooner or later, the competitively superior one will exclude the competitively inferior one, is what it says. So here they have not directly asked you for what is competitive exclusion principle, but they have given state the principle that explains elimination of one species. So you should immediately understand that they are talking about competitive exclusion principle. Moving on to second part of the question. Two different species can compete for the same resource. Give an example for it. Another example for competition where two different species are competing for the same resource. You can take the example of visiting flamingos and resident fishes in Lake Victoria. So both of them are dependent on zooplanktons for their food. So both of them are competing for zooplanktons. They are both two entirely different species. So that is one example that you can give in this case. How do species avoid competition in nature? Explain with an example. They do so by resource partitioning. Example for that you can give MacArthur's warblers. There were 5 species of warblers. 5 species of warblers. 5 species of them which existed on the same tree. They coexisted. Whatever resources were available, they shared it among themselves. They had different feeding habits. They chose different times of the day for foraging, and somehow, even though there are 5 different species competing for the same resource, they somehow managed to coexist. You can write about that as an example. Moving on to the next question. A student performed some crosses in plants and represented the result in the form of a bar graph as shown. Each graph displays the phenotypic proportion of the progeny. Study the graphs and answer the questions. So here you have 3 crosses. This is cross 1. Here you have cross 2. Here you have cross 3. The tall one is represented by blue color. The dwarf is represented by white color. Okay. So if you look at the cross, first cross, 100% of them are tall. If you look at the second cross, you have 75% tall and 25% dwarf. If you look at third cross, you have 50% tall, 50% dwarf. So let's see what are the questions that are following this. Option A, what can you infer about the genotype of parents in crosses 1 and 2? In the first cross, there are 2 possibilities. Either both the parents are tall, or one parent is homozygous tall, the other parent is homozygous dwarf. In either of these cases, you will get 100% tall plants. What about cross 2? We've got a 3:1 ratio in phenotype here. So this indicates that these 2 plants that are crossed are both heterozygous. Only in that case you will get a 3:1 ratio, right? This is what happens in F2 generation of monohybrid cross. Now here, 50%, 50%, 1:1 ratio. When do we get this? Whenever a heterozygous plant is crossed with a homozygous recessive plant. This is more like a test cross. In a test cross, you get 1:1 ratio, right? So here you can write for question 1, what can you infer about the genotype of parents in crosses 1 and 2? I have given you what is the genotype in cross 1 and 2. Which genetic cross is represented by these crosses? So it is monohybrid cross. Looking at the bar graph of cross 3, identify the type of cross performed and its importance in genetics. The cross that is performed here is a test cross. What is the importance of a test cross in genetics? So whenever you're not sure about the genotype of a organism showing the dominant phenotype. In order to identify whether that plant is homozygous or heterozygous, we perform the test cross, right? So here, this was a test cross result, monohybrid test cross. See, what conclusion can you draw from the results of the bar graphs of crosses 1 and 2? Name the genetic principle illustrated. So maybe you can talk about law of segregation here. Okay, so that is what you should be explaining in option C, because this is based on monohybrid cross, right? So here, since even though the parent has two different alleles, dissimilar pair of alleles or same alleles, eventually the gamete will receive only one of the two. That is what you will have to explain. I hope you have done that. Okay, so B part of the question, what are biofertilizers? Whenever you are using living organisms to enhance the nutrient quality of the soil, you call them biofertilizers. Name different types of microorganisms used as biofertilizers in organic farming and explain how each contributes to soil fertility. The different types of microorganisms that you use are, you use bacteria, we use cyanobacteria, and we also use fungi. Examples of bacteria includes rhizobium, which is symbiotic, azotobacter and azospirillum, which are, these both are free living nitrogen fixers. This is a symbiotic nitrogen fixer associated with roots of leguminous plants. Cyanobacteria also help in nitrogen fixation. You have nostoc, annabena and oscillatoria. So both of these help in nitrogen fixation. Fungi example, you have glomus. Glomus helps the plant absorb nutrients and water better by forming mycorrhizal associations. Specifically, it helps in enhancing the absorption of phosphorus from the soil. Write any two advantages of using biofertilizers over chemical fertilizers. Chemical fertilizers causes, it will eliminate even the beneficial pests from, fertilizers. So this is, okay, I thought it is, okay. Biofertilizers will make sure that the soil and the water, groundwater is not getting polluted. Since they are not chemicals, if any organism consumes these plant products, then other animals or humans, our life or our health will not be affected. Okay, so it does not contaminate the soil, it does not pollute the soil, it does not pollute the groundwater. Also, it is a safe option because we are not using chemicals, and chemicals sometimes can harm the health of the animals that feed on it. In this case, it does not. Okay, so that is the two advantages. Describe how lac operon operates both in the presence and absence of inducer E. coli. For this, you should have added, you should have drawn that entire lac operon thing, where you have promoter, repressor gene, promoter, operator, lac Z, lac Y, lac A, right? So this will always keep producing a repressor protein constitutively. This repressor in the presence of lactose will bind to lactose and get inactivated. When it is inactivated, the operator site is free, RNA polymerase comes in, binds to promoter, transcribes this polycistronic gene. It will eventually get translated and produce the three enzymes, beta-galactosidase, permease and transacetylase. So that will help in the metabolism of lactose. Whenever lactose is absent, lactose is present, that is the case. Whenever lactose is absent, what will happen? This repressor protein will go and bind to the operator and block the pathway for RNA polymerase. So RNA polymerase comes and binds at the promoter, but it will not be able to move ahead and transcribe the structural genes and translate it, which is when the operon is said to be switched off. Okay, so that is something that you would have had to explain in that case. Differentiate between Mendelian and chromosomal disorders. Mendelian disorders follow Mendel's laws. Chromosomal disorders, they are inherited differently. In Mendelian disorders, only one gene will be affected. In chromosomal disorders, it's usually the change that has happened at the chromosome level. Either the number of chromosomes is more or less than normal, or a part of the chromosome is broken, or there is a different type of chromosomal aberration. Because of what happens at the chromosomal level is what chromosomal disorders are. Because of what happens at nucleotide level, because of those kind of single gene mutations, we see Mendelian disorders. Another example you can give, another difference you can give examples. For Mendelian disorder, phenylketonuria, thalassemia, sickle cell anemia, color blindness, hemophilia, all of these are Mendelian disorders. For chromosomal disorders, Turner syndrome, Downs or Klinefelter's, any of those you could have mentioned. Which disorder is known as an inborn error of metabolism? This is phenylketonuria. Mention its cause. It is caused because one gene is mutated and that gene, which was supposed to produce an enzyme called phenylalanine hydroxylase, I will write PAH in short. This gene will not get produced. This gene's function is to convert phenylalanine to tyrosine. This will not get converted because this enzyme is not produced. So phenylalanine will instead get converted to phenyl pyruvic acid and other derivatives. So this will start getting accumulated in the body, gets accumulated in the brain, and some amount of it can be excreted along with the urine also. So that is the case of phenylketonuria. Mention any one symptom. One symptom you can write mental retardation, reduction in skin pigmentation, reduction in hair pigmentation, any one of those you could have written. Next, name the harmful compounds it forms, phenyl pyruvic acid and other derivatives you can write. Okay. Now, moving on to our next question. Just a minute. Wait, is it done already? Are we done with the botany portion? Let me quickly check the paper. Just a minute. Let me know in the meantime in the chat, how was the paper? How did you perform? There is some Chinese agent in our chat. What are you doing, Chinese guy? Yeah, Arushi Singh is here. Good job, Arushi. Congratulations. I wasn't able to see the chat until now. For some reason, it was stuck. I'm able to see it now. Just give me 1 minute and I'll confirm if I am done. Yep, we are done with the botany portions, and Vishwajit sir will discuss the zoology sections. Just a minute. Okay. This is set 3, Piyush. Piyush Gupta, this is set 3. Did you find any question particularly difficult? Like, did any question give you like a panic attack? The questions were as expected, like they're not straightforward. Obviously, in your board exams, you don't get straightforward questions. Some of them are like straightforward, but some of them are twisted. For as long as you had conceptual clarity, I'm sure you would have answered very well. Now, I will end this and Vishwajit sir will take over. Okay, bye-bye. See you. Okay, Hansika, I'm glad it was helpful. We're also going to be starting a series for NEET on the PW NEET English YouTube channel. It's a free series. It's going to start from the 31st. So, yeah, in case you're NEET aspirants, you can come join us. We'll be discussing questions every day, probably at 6 p.m. in the evening. We will cover your entire syllabus, MCQs. Okay? So, if you haven't yet subscribed to PW NEET English YouTube channel, this is your time to consider that if you're serious NEET aspirants. It's not going to happen on this channel. There's another channel called PW NEET English. So, look it up and subscribe and join our sessions. Malayalam, I don't know. Malare unnai, that's all I know. All right, bye bye.
So, very good evening or afternoon, everyone. So, how you all are doing? Hope after the completion of your biology examination for CBSE 12th board, few of you or most of you are feeling stress is released. So, I would like to know that what's your experience for today's examination? Was it moderate, easier, or tough? Right? The set of question I'm having, that is set 3, and according to me it should be easier towards moderate for particularly zoology section. Okay, so let's get started with first question in zoology section, and that is, the question is very easy. Interference are most effective in making non-infected cells resistant against the spread of which of the following disease? We know that interference will be acting as a biological response modifiers, right? So the answer is it will be acting against cancer. So this is our answer. Though we know that cancer has no answer, but for today's session we got the particular answer for this particular question. Now let's check the next question, and the question is, select the option that shows correct statements. Okay, so let's check them one by one and try to solve it. Okay, yeah, same here, Chitra. Okay, so we in our chat section we got someone from China as well as Chinese agent is among us. Okay, so corpus luteum secretes progesterone. Exactly, it's a temporary endocrine gland inside the ovary. Only FSH attains a peak level in the middle of the cycle. False. We know about LH surge. Okay, so LH surge induces rupture of Graafian follicle. Again, that's a correct one. There will be 10 times more tighter concentration. We'll be finding that LH is present into the maternal blood just prior to the ovulation. So that is called LH surge. Now, fourth option is saying luteal phase of menstrual cycle is also called proliferative phase. Wrong one. Luteal phase is called as secretory phase. Proliferative phase, the second phase of menstrual cycle. So according to our question, option 1 and option 3, they are correct one. Option 1 and option 3 for this particular question. Moving ahead. Okay, now let's check out this particular question. Which connective tissue connects ovary to pelvic wall and uterus? If you have studied your NCERT, it's written there, obviously ovarian ligaments. Yes, if we talk about uterus, so this is uterus and the ovary is present both the side and it's attached with the help of uterine ligament or ovarian ligament. Now let's move forward to the next question. Expression of a particular gene help us to identify transformants. What is this gene known as? Okay, the question is from the chapter biotechnology and its biotechnology principle and processes, and the expression of a gene if that will be helping us to identify the transformed bacterial colony. We call that as selectable marker. Sometimes they can be called as reporter gene. Yes, and we know that they are like ampicillin resistant gene, tetracycline resistant gene, kanamycin resistant gene, as well as sometimes LAGZ also act as the selectable marker or reporter. Next question. Which of the following evidences does not support Darwin's theory of natural selection? Okay, Darwin tells us about branching descent. The evolution described by Sir Darwin is small and directional, gradual, very slow. So that's perfect explanation of Darwinism. Appearance of new traits by mutation. No, mutation was described by Hugo de Vries. And survival of the fittest, again one of the important statement given by Sir Darwin. So we got our answer. Does not support Darwinism, that is appearance of new traits by mutation, or the saltation theory of evolution was given by Hugo de Vries. Yeah. Let's move forward to the next question, and here I can see assertion and reason. And the assertion is saying fossils provide direct evidence for evolution, and the reason is saying obviously that's a true statement. Reason is saying fossils help to trace evolutionary relationship among organisms over time. Yes, here the reason is talking about biogeological time scale. Okay, if you are excavating the earth crust layer wise layer while you are going down in a particular set of rock will be displaying fossilized forms or rockified forms of some particular organisms. So we can say that reason is justifying the assertion. So the answer is option A. Both assertion and reason are true, and reason is the correct explanation for the assertion. Now let's move forward. Pomato, again another question from biotechnology, was produced by fusing protoplast of, very easy question, and yes, the fossils will be getting the layers of sedimentary rocks specifically. Okay, now pomato is a fusion of protoplast of two plants, tomato and potato. So this is our answer. Again, it was a very easy one. Now another question. Health officer visiting two different regions and reported following symptoms in patients. Okay, another question from Human Health and Disease, and the region A. You can observe the symptoms: sustained high fever with stomach pain, constipation and loss of appetite. Yes, direct line from NCERT, and the disease is typhoid, right? So for region A, the disease is typhoid, and the causative agent is a gram-negative bacteria called Salmonella typhi. Let's check. Let's move to the region B. Now here you can observe inflammation in the lower limbs and genital organs. Yes. So that particular patient of region B affected by Wuchereria, and the disease, that is filariasis. Filariasis, or you can call it elephant foot disease or elephantiasis, and caused by Wuchereria. Wuchereria, two species are mentioned, Wuchereria bancropti and Wuchereria malai. Yes, typhoid is by Salmonella typhi. Yeah. Yes, exactly, Sidhu. So name the diseases like affecting the patients in the regions A and B. Obviously, we have discussed typhoid and filariasis, and write down the scientific name of the causative agent. One is Salmonella, another is Wuchereria. Okay, we are done with this. Let's move forward. Another assertion reason. Okay, so that means CBSE as well as NTA, they really like this particular kind of question, assertion and reason. Okay, those who are reading the NCERT in a proper manner, it's very easy to answer these kind of questions called assertion and reason. And the assertion is saying the milk produced by transgenic cow Rosie was nutritionally more balanced product for human babies than natural cow milk. That is true. And the reason is said that it was human protein enriched milk containing human alpha lactalbumin. That's also true. So why the milk of Rosie is more containing more nutrition than in compared to natural cow milk? Because it's containing alpha lactalbumin. So here the answer will be both the assertion and reason, they are true, and reason is perfectly explaining the assertion. Yes. Next question, what is adaptive radiation? Give one example from any habitat. Okay, so adaptive radiation means if there is a particular geographical zone, suppose a bird species is staying here into this particular geographical zone, right? Suppose it's finch bird. Now due to some reason, in search of food, in search of better breeding ground, or due to climate change or natural calamity, few individuals in a scattered manner, they have established their new colonies into different islands. So when a particular population, individuals from a particular population is scattering or spreading throughout different geographical zones, that is called radiation, and they will be adapted accordingly depending upon their need, feeding habit and the climatic condition. So here the example is Darwin finches. Darwin finches. Okay. Fingers, no, Darwin finches, yes. They're classical example of adaptive radiation. So the native finch of uh, Galapagos Island, they had very heavy beaks, right? Now, when they were dispersed into different smaller islets or smaller islands, few birds, few finches, they got tiny and smaller beaks. Few birds, they got heavier beaks to crush the nuts or the seeds, and the smaller beaks adapted to catch the insects. So depending upon their feeding habit, there was acclimatization and adaptation in course of time for this finch bird into different islands or islets. Next question. Observe the given picture carefully. A mixture of DNA with fragments ranging from 100 base pair to 1800 base pairs were separated by electrophoresis on agarose gel. So with the following arrangement, so you can check the arrangement, the diagram already given in the question paper. Okay. So first question, what results will be obtained in staining with ethidium bromide? Explain with reasons. Okay. So if you are staining the DNA molecules with ETBR, ethidium bromide, they will act as chelating or intercalating agent, and if you are observing them with UV rays, then they will produce fluorescence and bright orange light. They will create a fluorescence of orange color, and under ultraviolet radiation you can really observe them, because normal DNA molecules, they are not really visible under UV rays. Yes, bright orange color. Now, the above setup was modified as shown below, and a band with 100 base pairs was obtained at X. So just you can check, the polarity is been changed. So towards the wells inside which we put DNA molecules to separate them based upon their size, right? So towards the well it was positive electrode, far from the well it was negative electrode. But the second scenario you can observe, towards the wells it's negative electrode, and the farther side we got the positive electrode, and when the circuit is closed, gradually the DNA will start moving, and you know this X band is running very fast towards positivity, because it's the smallest among the DNA molecules. So the X band of this DNA denoting towards the smallest base pair of DNA, it's 100 base pair DNA molecule, smallest DNA molecule among, because the range was in between 100 to 1800. So this particular bands, the DNAs are of 1800 base pair, and this particular band is of 100 base pair. Okay, what changes were made in the previous design? Altering the polarity, to get the X band, and X band that is appearing as X, that is of 100 base pair DNA. Now let's move forward to the next question. Identify the stage of follicle where primary oocyte undergoes first meiotic division. Also mention the products of this division. So to identify the stage of follicle where primary oocyte undergoes first meiotic division, yes, for that we have to track the follicle. If we check primary follicle, inside primary follicle the stage of the immature ovum, that we will be finding that is primary oocyte only. Then the developed version in compared to the primary follicle, that is secondary follicle. Even inside secondary follicle, the version of the maturing egg that we will be finding, that is secondary oocyte. Sorry, that is primary oocyte again. That is primary oocyte again you will be finding inside secondary follicle. Now gradually the secondary follicle will be modified with tertiary follicle forming an antrum, and inside this, while the secondary follicle is modifying into tertiary follicle, the primary oocyte gonna undergo or complete the first meiotic division and it will become secondary oocyte. Okay. So we have to identify the stage, and the stage is tertiary follicle. The stage is tertiary follicle, and also mention the products of this division. Okay. So products of this division will be, from primary follicle there will be formation of a smaller cell and a larger cell. The larger cell is called as secondary oocyte, which is haploid in nature, and the smaller cell is called as first polar body, again which is haploid in nature. Now why there is equal karyokinesis but unequal cytokinesis? Because the primary oocyte that is devoid of a crucial cell organelle called centrioles, and that is the reason there will be equal nuclear division but unequal cytoplasmic division. That's why the resultant cells, one is bigger, one is smaller. Okay. Now you have to mention the scientific name of the source plant and the part from which opioids are extracted. Now opioids means morphine, heroin. These are considered as opioid drugs, and the source plant is Papaver somniferum. Simply we call these as poppy plant. Okay. From the latex, the milky fluid of this poppy plant, we can obtain these opioid drugs. Okay. Next question, how morphine and heroin are related? So simply, if you are getting morphine with you in laboratory, you have to do acetylation. Acetylation. Now upon acetylation of morphine, you are gonna get diacetyl morphine, and this diacetyl morphine got another name, that is heroin. Simply in layman tongue we can call these as smack. Okay. Now mention the effects each one of them has on human body. Okay. Now sometime we use morphine as painkiller. Sometimes we use morphine as painkiller just after surgery, right? And this heroin or sometimes overdose of morphine can act our central nervous system as well as gastrointestinal tract, because opioid receptors are present into these particular organs. if you are taking opioid drugs, they will be interacting with your body with the receptors present into your central nervous system and gastrointestinal tract, and they will get inside your body and affect your nerve mechanism or nervous system. Nerve impulse conduction will be impacted, and you will get a calmness, a dizziness. That's why these opioid drugs are called as depressant drugs. Okay, they can interfere even with your nerve impulse conduction, causing depression, causing calmness. So next question is, how Hardy-Weinberg expression, that is p square 2pq plus q square equals to 1 was derived. Okay, so according to the Hardy-Weinberg equation, we know that allelic frequency of a population is always constant, that is p plus q equals to 1. Okay, p is the frequency of homozygous homozygous dominant alleles, and q is the frequency of homozygous recessive allele. Okay, now we have to do binomial expression. If you are doing binomial, if you want to get the product of binomial expression, you have to do square both the side, and the result you will be getting here, p square plus 2pq plus q square equals to 1. Now, if you want to tally this with Mendelian experiment from monohybrid cross, you can obtain the similar kind of result. Like monohybrid cross, we have taken tall pea plant capital T into small t small t. Then after the cross hybrid or hybridization or crossing in between them, we got the F1 generation, that was heterozygous dominant individuals, that is first filial generation, and upon selfing in between them we got F2 generation, and the genotype is like this: capital T capital T capital T small t capital T small t small t small t. So here the heterozygous dominant individuals, sorry, homozygous dominant individuals, their capital T, that is 1 in ratio. Okay, heterozygous dominant individuals 2 capital T small t, that is 2 into ratio, and homozygous recessive condition small t, that is 1 in ratio. Now accordingly, we know that capital T is representing the dominant frequency of dominant allele, small t is representing the frequency of a recessive allele. Capital T is P and small t is Q. Now if you put it here accordingly, so what you will be getting? Capital T equals to P square, P into P. Okay, now here we got capital T, 2 capital T small t, so it can be written as 2PQ, and the small t can be written as Q square, and overall if we merge them the frequency will be 1. So from this particular equation also you can prove the binomial expression of Hardy-Weinberg equilibrium. Yes, that was alpha human lactalbumin. I think question was alpha human lactalbumin was there. Yes, here the question, the reason is saying the human protein enriched milk containing human alpha lactalbumin. So this is a true reason. Okay, now question B from this particular question: list any 2 factors that disrupt the genetic equilibrium. Okay, genetic equilibrium can be disrupted by genetic drift, it can be disrupted by mutation. because in both the cases there will be change in allelic frequency. Next question. Name the protozoan species that is responsible for causing the most serious and even fatal malarial disease. Okay. So, the protozoan species is Plasmodium falciparum. Plasmodium falciparum is responsible for causing malignant malaria. Okay. Name the host in which the parasite completes its sexual stages and explain the changes taking place. Name the host in which the parasite complete the sexual stages. Sexual stages will be completed into female Anopheles mosquito. And the changes there are taking place, gametocytes will enter into mosquito body. Gametocytes will enter into the mosquito body, and inside the gut of female Anopheles mosquito, the gametocyte will be converted into gametes, into sperm and ovum. Fertilization will take place inside that gut of mosquito only, and upon fertilization they will form a motile embryo, and that motile embryo will enter into the body wall. That motile embryo will enter into the body wall of that mosquito. Now the embryo will stop moving into the body. Now we will be calling that as oocyst. Now within few hours that oocyst will perform sporogony, means spore formation will be there, and there will be formation of thousands of sporozoites, and these sporozoites will try to escape and enter into the salivary gland of that infected mosquito. Okay. Now next question. How does the parasite damage the human body after entering the blood stream? Okay. So from blood it will search for the liver, and these hepatocyte cells of liver, they are nutrition provider for the sporozoite stage. So sporozoite stage will enter into the blood and then it will attack liver, and upon damaging the liver cell it will come out and it will attack RBC. Okay. Now when the RBC is damaged, it will release a toxin called hemozoin. Due to the influence of this hemozoin toxin, you will suffer from fever. So that's how the malarial parasite will be damaging our body. First it will attack liver cells, hepatocytes, then it will attack RBC. Suggests two effective preventive measures to control the spread of the disease in endemic regions. Mosquito repellent can be used, or sometime in stagnant water body or drainage systems, larvivorous fishes like guppy and gambusia can be introduced. Okay. Now the next question is, trace the events from fertilization till the implantation of the blastocyst in the human female. Also maintain the site where fertilization takes place. Okay. From fertilization to implantation, we have to explain. So if this is the uterus and this is the fallopian tube, suppose sperm is already entered into the ampullary region, ampulla, according to NCERT, fertilization will take place into the ampullary region, and after fertilization there will be formation of zygote, diploid single diploid unicellular embryonic stage, that is called zygote. Okay. Now to remember this, the zygote is covered by zona pellucida even after fertilization. The outermost layer of the zygote is called zona pellucida, and it will start development called as zygotic cleavage. Though zygotic cleavage is technically mitosis, but the cells are not maintaining their size, they are reducing their size. That is why instead of saying simple mitosis, we call the division of the zygote as zygotic cleavage. So gradually the cell number will increase, 1 cell to 2 cell, 2 cell to 4 cell, 4 cell to 8 cell. So gradually the number will start increasing, and when the embryo is reaching at the stage of 8 cell stage, embryo is reaching at 8 cell stage, that is called as morula. Now 8 to 16 cell stage or 8 to 16 blastomere stages of the embryo, we call it as morula because it looks like mulberry leaves. Okay. So this is how the embryonic stages will progress. Upon fertilization there will be formation of zygote. Upon zygotic cleavage there will be formation of 2 cell stage, then 4 cell stage, then 8 cell stage, then 16 cell stage. Now these 8 cell and 16 cell stages, they are called as morula stage, and the initial cells are called as blastomeres. Initial cells of the zygotic development or embryonic development, initial cells are called as blastomeres. Now these blastomeres, they can be called as totipotent or pluripotent cells. Now gradually the embryo is coming downwards into the uterine cavity. Now this uterine cavity is highly receptive for to the upcoming downcoming embryo, and the innermost layer of uterus, that is called as endometrium. This is the place where the blastocyst stage will get implanted inside the uterus. So the stage which is getting implanted, that is known as blastocyst stage, and just prior to the implantation, the zona pellucida is ruptured. Okay. So why the embryo is maintaining zona pellucida up to the implantation? Because the zona pellucida is acting as the deciding factor where exactly to implant that developing embryo. That's why just prior to the implantation, the actual embryo is coming out of the zona pellucida covering, and the phase is called blastocyst. And why this phase is called as blastocyst, if you check it properly, a tumor or cyst kind of inner cell mass is found, and it's a hollow cavity there is a hollow cavity inside we call it as a blastocele. So it's a football kind of shape, football kind of structure of embryonic developmental phase, and that's why we have given the name of this particular embryonic stage as blastocyst. Sometimes we call it blastula. (16). Okay, let me check the question number 16. Okay, 16 is in not in my section. I'm discussing the questions of zoology. Okay. So we are done with this particular question. Now let's follow the next question, question B. The case studies of some couples are given below. They were not able to have kids even though the parents were not taking any precautions. So it's a case of infertility. They also do not wish to adopt a child or take the help of donor. Okay, study this case and suggest an appropriate assisted reproductive technology that could help them. Okay, so first case is like this, and the female is normal, female is having normal report. Okay, and the male can produce normal sperms, but no connection between the epididymis and vas difference. Okay, but no connection between the epididymis and vas difference. Okay, so the secondary or accessory reproductive duct is somehow damaged, but that male can produce normal sperm. Okay, so in that case the normal sperm can be collected from the epididymis. Normal sperm means structurally and functionally matured sperm can be collected from epididymis, because structural maturation of sperm takes place inside the testes, inside the seminiferous tubules, and the functional maturation of the sperm takes place inside the epididymis. As because the connection is intact up to epididymis, and after epididymis the reproductive tract is somehow torn or broken or damaged. So we can collect the sperm from the epididymis. You can collect the sperm from the epididymis, and we can take the matured or secondary oocyte from that female, and we can go with ICSI, intracytoplasmic sperm injection. Intracytoplasmic sperm injection is prescribed here, as because you are collecting raw sperms from epididymis, you are not collecting the seminal plasma, there is no semen. That's why artificial insemination is kind of tough for this particular case. Okay. Next, case 2. Female is producing normal eggs, but fallopian tube is blocked. Okay. Male can produce normal sperms, and male is also reported as normal. Okay, so here we have to go with in vivo fertilization, and the technique will be prescribed here, that is gift gamete intrafallopian transfer. Okay, the normal egg is produced by the female. Okay, sorry, normal egg is produced by the female, but fallopian tube is blocked. Okay, so we will be collecting the egg from the female and we will be putting into the uterus. So we will be performing intrauterine transfer. Uterine transfer or GIFT can be also performed. Gamete intrafallopian transfer will be more reliable here. The female is producing normal eggs, but fallopian tube is somehow blocked. Okay. Now case 3, female can produce, okay, female is having normal report, male low sperm count with less motility. So in this case, will be performing intrauterine insemination, IUI. Okay. So this is how, this was the paper of zoology. Now any other question if you want to ask, you can mention in the comment section. Okay, the things were in very much hurry. So if there any discrepancy, you can mention in the comment box as well. Okay, I will recheck that and I will discuss with you. Yeah, someone was commenting that finches are not found in Galapagos Island, and he was laughing like anything. So what's wrong in that? Finches are primarily found in Galapagos and the nearby islets only. Okay, anything more? Okay, so if you don't have anything to discuss here, then we can end the session. Usually saying lactoglobin is written. Let me check. Lactoglobin or lactalbumin? Should be lactalbumin. Let me check again. Okay. Twice I have overlooked that. Yes, you are absolutely correct. So it's lactoglobulin. It's written here. Okay, as because it's hazy in my board here, it's not very clear. It's lactoglobulin. So this is wrong. Yes, it should be lactalbumin. So in that case, the answer will be assertion is true and reason is false. Okay. Mm, assertion is true, but reason is false. In that case, the answer will be option C. Thank you very much, Ujjwal. Okay, I just overlooked that. Twice I was reading that as lactalbumin, but actually it's written as lactoglobulin. Question number 16. Question number 16, I think that is in botany section. What is the question? Can you please mention the question number 16, what you were asking for? I think question number 16. Okay, flocks are masses of bacteria associated with fungal filaments. In secondary treatment of sewage, flocks help in digestion of solid waste by anaerobic respiration. Okay, yes, flocks will be digesting the solid waste by uh, aerobic respiration. Yes, option third will be the correct. Yes. Okay, he's talking about this question. Yes, exactly. It was my somehow I have overlooked. Yeah, you are right. Okay, I read that as a lactalbumin. Okay, thank you for pointing out that particular point. Okay, if anything more, uh, Okay, let me check again. The art. So if female is normal, female is having normal report, the male is producing normal sperm but no connection between epididymis. Yes, in that case we can perform ICSI, intracytoplasmic sperm injection, right? That's quite reliable method. And next question, if female can produce normal egg but fallopian tube is blocked. Fallopian tube is blocked means in normal way that the passage is blocked for the secondary oocyte to come inside, but we can perform gift in that case. Fallopian tube is not damaged here, fallopian tube is blocked. Normal sperm, male reports are normal. Gift can be performed here. Some other thing we can perform uh, here also. For case 2 also you can perform ICSI, intracytoplasmic sperm injection is the easiest way. For most of the cases cytoplasmic sperm injection can be performed. Apart from that, uh, zygote intrafallopian transfer. Okay, so zipped will not be that much reliable. Intrauterine transfer for case 2 you can also go for intrauterine transfer if you are going with ET. If you are going with embryo transfer, then you can go with intrauterine transfer as well. For case 2 there are multiple options are coming with us. Okay, and case 3, female is having normal report and the male is having low sperm count, means oligospermia with less motility. Okay, so in that case normal artificial insemination cannot be done. In that case we have to go for intrauterine insemination. If there is low sperm count with less motile sperm, yeah, intrauterine insemination will be working in this case 3, right? So that was all. Aesthetic me core. Okay, if you have something contradictory against the answer, you can express yours, right? So what do you think for that fallopian tube? If fallopian tube is blocked, then what we will go for, with which process of art? Aesthetic me. Okay, no issue. uh, you can like write your comment. You are free to write comment and free to discuss again. And if you don't have any more thing to ask or any more thing to discuss, then I'm ending the session. And best of luck for your future and those who are preparing for NEET. So the stress is not relieved yet. Till you have nearby 30 to 35 days left, and you have to work upon your NCRT again with in a rebrashed manner. Okay, so bye bye. Thank you. हाँ इन करवा हो गया हाँ 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 इन कर दिए